Diagnoses for autism spectrum disorder appear to be more frequent than ever before, and genetics research has revealed some compelling influences for the increase in prevalence. Today, I'll be discussing 3 genetics concepts and how they contribute to the symptoms of autism spectrum disorder. Your DNA is like a cookbook, and certain DNA sequences are like recipes used to create specific dishes, or in this case, proteins that make up every part of your body, like your bones, skin, and other organs. Within the DNA structure, copy number variations are large repeated segments of the DNA, but they can vary in how many copies are missing or present. This is similar to having multiple pages of a recipe in a cookbook, or discovering that some pages for a certain recipe are missing. In the context of genetics, variations in the presence of repeated segments of DNA can affect important chromosomal regions. For example, chromosome 15 has regions that code for genes that control synaptic signaling and neural inhibition. If these regions are duplicated, it could result in imbalances in excitation and inhibition in the brain, a common symptom within autism spectrum disorder. In chromosome 16, there are also important regions of DNA that code for genes involved in brain development and neuronal signaling. If these regions of DNA are deleted, the genetic material that codes for the necessary proteins won't be available. This can result in underdeveloped brain structures and other symptoms displayed in autism spectrum disorder, like language and speech difficulties or motor dysfunction. Transposable elements, also known as jumping genes, are another factor in genetics that can contribute to the prevalence of autism spectrum disorder. To transpose means to change different things to a different place or context. These jumping genes are sequences of DNA that can move to new locations within the genome. They can disrupt nearby genes or influence how those genes are turned on or off. Back to the cooking analogy, if DNA is like a cookbook, then transposable elements would be if certain ingredients showed up in the incorrect recipe. You wouldn't be able to properly make the intended dish. Transposable elements make up about half of all the elements of your genome, the entire cookbook of proteins used to make up your body. But many of them are inactive, located in non-coding regions, or are checked by epigenetic modifications. While some transposable elements are inactive, some remain mobile, especially during early development or in brain cells. Their movement can disrupt important genes or regulatory regions linked to neural function, explaining the rise of neurodivergence and more symptoms observed in autism spectrum disorder. Lastly, we have mutations, which are irreversible changes to the genetic sequence. Mutations can be particularly troubling because they cannot be undone, although they may potentially be harmless. In our analogy, mutations could be a number of inheritable changes that permanently change recipes by changing, inserting, or deleting ingredients. Some of these mutations can be directly inherited by offspring, but de novo mutations are a classification of mutations that occur spontaneously within a sperm or egg without being inherited from a parent. In the context of autism spectrum disorder, mutations in genes responsible for brain development could alter the proteins that are transcribed from a given genetic sequence. Mutations in these genetic regions could explain more of the underdeveloped brain structures and connectivity that cause the observable symptoms in autism spectrum disorder, like specific interests, speech pathology, repetitive behaviors, or difficulties socializing.